Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be doing a review for Biology 2421 Practical number 2. So here are the notes I had written on the board for Helminths. You can see I wanted you to know about the major phyla nematodes, which are the roundworms that can usually form cysts, versus the Platyhelminthes, which are known as the flatworms. Let's start with the nematodes. The example species I wanted you to know about were Ancylosoma, which is the hookworm, Dirofilaria, the heartworm, Trichinella, pork roundworm, and Enterobius, which is the pinworm. With Platyhelminthes, there were two major classes to know about: trematodes, which are the flukes, and cestodes, which are the tapeworms. Let's start with trematodes, the flukes. The two major examples of flukes I wanted you to remember and be able to identify are Fasciola, the liver fluke, and Schistosoma, the blood fluke. With Schistosoma, remember, a lot of times we might show you the image of the mating between the slender female fitting along the groove on the shorter, wider male. I'll show you an image of that, and I believe I have an image of that, or at least I have an image of schistosoma. I'll show you in a second. With regard to the tapeworms, you know, the cestodes, we need to know that they are hermaphroditic, so they have both male and female gamete formation. And the example species here is Taenia. Taenia is the typical tapeworm. The body has a head. Right, the head is called the scolex, and you should know that the scolex has hooks and suckers, while the body proper itself is made up of segments called proglottid. Let me go into the lab manual now and show you a little bit more detail about these worms. So again, this is your module on Helminths. Make sure to read through this. You don't need to know how to identify eggs or anything like that, but you should know about the liver fluke here. You should know about flukes, fasciola. This is schistosoma. Remember, we might show you an image of that mating. This is Taenia, the tapeworm. Here is the sucker. These are the hooks. These are the suckers here. These are the proglottic body segments. Next, we have the pinworm here. Please be able to identify that under the microscope. The pinworm's head looks like a pin. Then you have trichinella. Trichinella, which forms cysts. This image here is pig muscle, so like pig meat. And what you're seeing here, these little spirals, these are the cysts of trichinella in the pig tissue. Dirofilaria, this is heartworm, though usually we don't ask you about Dirofilaria. Okay, so please be able to identify these under the microscope. All right, now with regard to the yogurt lab, recall that it was Streptococcus thermophilus as well as Lactobacillus vulgaricus that were the two major species of bacteria for lactose breakdown in the milk, producing the yogurt. Other than that, there's really not much more meaty material here. Why would you have to cool the milk to about 45 degrees before adding starter culture? Well, because obviously starter culture will die any warmer than 45 degrees C. This is too warm, and it could denature the proteins in the starter culture bacterium and kill the starter culture. All right, with regard to the unknown identification lab, remember that you were handed a bacteria of the family Enterobacteriaceae. These are the enteric bacteria, which are gram-negative rod. They are facultative anaerobes, and a lot of them are gut bacteria. That's what enteric bacteria means. And remember, we did a number of tests over several class periods to identify the unknown. So be familiar with all of these tests listed here. Let's go through a few, starting with the IMViC tests, which include the SIM test. Let's go ahead and talk about those. All right, recall for the IMViC tests, one of the tests was called the SIM test. This is an indole, motility, and hydrogen sulfide test. It has 3 tests in one. It checks for motility, if possible. Remember, unless it turned black due to H2S production, in which case it's impossible to check for motility. You can check for motility. You can check for H2S, hydrogen sulfide, which appears as a black precipitant. And by adding 6 to 8 drops of the Kovacs reagent, a hot pink color change is indicative of indole production. So you should know all of these things. The SIM test is a motility test, a test for H2S, and a test for indole production. And you should know that the reagent is called Kovacs reagent. Additionally, you should know what the acronym IMViC stands for. Remember, you have indole, methyl red, Voges, and citrate. Remember the citrate slant? Let's talk about that next. It was a slant that started out as a green-colored slant. This indicates citrate use, if your bacteria can use citrate or not. Citrate use results in an alkaline product. The color indicator is bromethymol blue, which changes color from a green to a royal blue to indicate citrate use and to indicate an alkaline product. So again, if it turns royal blue, this is citrate positive, and you should know that the color indicator is bromethymol blue. Next, we had the MRVP tests. Remember, you had an MRVP tube. You poured 1/3 of it into one test tube. This became your methyl red test, which detects glucose use via mixed acid fermentation. For the methyl red test, you added 6 to 8 drops of the methyl red reagent, and a red color change was indicative of glucose use with mixed acid fermentation. And if it remained yellow or kind of like a orangey color, that was a negative test for methyl red. So again, you should know that the methyl red test checks for glucose use with mixed acid fermentation. You should know that the reagent is called the methyl red reagent, and you should know that a red color change is a positive result. Then the remaining 2/3 of the MRVP tube was serving as the VP test. Remember, this stands for the Voges test, which detects acetoin. You should see, which detects acetoin. You should know you add 12 drops, or you don't need to know the exact number of drops, but you add Barrett's reagent A and then Barrett's reagent B. You have to let it sit for 20 to 30 minutes undisturbed, and if there's red, if there's red in there, even at the top, if it turns red, that's a positive result for acetoin, and yellow is negative. So again, you should know Barrett's A and Barrett's B are the reagents, and you should know it needs to sit undisturbed, and a red color change is positive. So those are the IMViC tests. All right, next, recall that we did oxygen requirement tests in 2 ways. We did a plate test where we streaked our unknown bacterium on 3 different TSA plates and then incubated either in air, just in the incubator, in a candle jar, or in a gas pack. Now, the air contained your standard atmospheric 20 to 22% oxygen. The candle jar depleted the oxygen down to 8 to 10%, and the gas pack completely depleted the oxygen down to 0%. The other test we did for oxygen was called the Thioglycolate broth test. Remember that thioglycolate is a reducing agent that removes the oxygen from the broth, and then we were able to look for where the bacteria grows in the broth to determine its oxygen requirement. So as you can see here, if there's growth at the top, this indicates that the bacteria is a strict aerobe. If there's growth only at the bottom, this indicates a strict anaerobe. If there's growth not quite at the top, at the surface of the solution, but slightly underneath, this is indicative of a microaerophile. And if the growth is throughout the tube, these are either aerotolerant or facultative anaerobic bacteria. Another thing is when you look at the results of your plates, remember, you could get questions like, is this a strict aerobe, a strict anaerobe, or is it a facultative bacterium? Remember, you would look at all three plates, and let's say your bacterium is A, the bacterium A here. So the species A. Notice how bacterium A grew on all three plates. If it grew on all three plates, that means it can grow with or without oxygen, so it would be a facultative anaerobe. However, bacterium B, notice that it only grew on the candle jar. It mainly grew on candle jar. It didn't grow very well in gas pack with 0% oxygen. It did not grow so well with ambient air. So in this case, it would be a microaerophile, right? Because it grew in low levels of oxygen. C, on the other hand, look at bacteria C. Bacteria C did not grow in air, did not grow in candle jar. It grew mainly in gas pack, so that would be your strict anaerobe. See, so you can use either a plate method for interpretation of oxygen requirement, or you could use the thioglycolate broth method for the same thing as well. All right, next we did the gelatin test, which checks for gelatinase. We did a needles poke inoculation of a deep, the gelatinase deep. If the bacteria does produce gelatinase, this changes the solid medium to a liquid one. Also, what you should know is that we had to place this either in the fridge or on ice for 10 minutes because the reason for that was to check for false positives. If the solution is still liquid after incubation in the fridge, then it is a true gelatinase positive or gelatin positive test. All right, next, the decarboxylase test. Do you recall the decarboxylase test? This checks for the enzyme decarboxylase. Just to remind you, amino acids look like this. They have an amino group, a carboxyl group, an alpha carbon, and a hydrogen, as well as a side chain. Decarboxylases remove the carboxyl group. You see this dashed red line. They remove the carboxyl group, leaving behind a basic product. So here we're checking for the enzyme decarboxylase for different amino acids. I believe there was a lysine decarboxylase, an ornithine decarboxylase, arginine decarboxylase, and then your no amino acid control as well. Here you should know that the color indicator is called Brom-Cresol blue, and you should know that what you're looking for is a basic product. Purple color is a positive result, and a yellow color is a negative result. Okay, and also recall that, remember, after we inoculated our tubes, we also overlay with oil, with mineral oil. Next, the deaminase test. Do you remember the deaminase test was actually a slant? And just like the decarboxylase cleaves the carboxyl group, the deaminase test, that we're checking for an enzyme deaminase, which would remove the amino group from the amino acid. So here we're checking for deaminase, and specifically for the amino acid phenylalanine, cleavage of this amino group results in an acidic product this time, called phenylpyruvic acid. How did we test for that? Do you recall after incubation we added a reagent called ferric chloride, or FeCl3, ferric chloride. And remember that an avocado green color shift was indicative of a positive result. So we were looking for an avocado color shift. Next, for the OF glucose test, we were looking for oxidative or fermentative carbohydrate use. Here, a negative result looks green, and a positive result for sugar use turns yellow. So here you can see yellow for both oxidative and fermentative. And in this example here, it only turned yellow at the top of the oil-free tube, so it's oxidative. Remember, one of the tubes had a mineral oil overlay, that's your fermentative tube, and the other tube did not have the oil overlay, that is your oxidative tube. So a result such as this would indicate oxidative fermentation, oxidative respiration, and a result such as this would indicate oxidative and fermentative, so it would be fermentative. All right, next for the starch test. Remember, for the starch test, we're looking for the enzyme amylase. The starch was already mixed into the agar plate. Then the bacteria grow on the plate. If the bacteria are able to produce amylase, then addition of iodine, the reagent iodine, will show that there is no purple color change near the bacteria. So the bacteria kind of have a halo of no purple color. Remember that iodine reacts with starch to form a purple precipitant, and if the starch is broken down due to the amylase, then the bacteria are going to have this clear area with no purple color change near the bacterial growth. So you should know that what a positive result looks like, and you should know that iodine is the reagent that needs to be added here. With the lipid test, okay, the lipid test, you're looking for lipase. The enzyme is called lipase. Lipase breaks down tributylin, which is vegetable oil, into fatty acids. Remember, this was your blue plate. It was blue because of addition of methylene blue as a color indicator. If the bacteria produces lipase, this breaks down tributylin into fatty acids. This also causes a breakdown in the methylene blue color, such that the bacteria have this clear zone around them, this kind of halo where the blue color is missing around the bacterial growth. Okay, and then lastly here, casein. Remember, this is called the skim milk plate or the casein hydrolysis plate. This tests for the enzyme caseinase. Casein is a major milk protein, and the plate has milk mixed in. So skim milk is mixed into the agar. That's why it has this kind of opaque white color. If the bacteria can produce caseinase, then the bacteria can break down casein into amino acids, and this gives you a clearing or a halo of no white color around the bacterial growth. So if you have a halo lacking the white color around the bacterial growth, this is indicative of caseinase. So let's take a quick break time with Gizmo and Wicket, and we'll come back to finish this review strong. For urinary tract culture lab, remember that less than 10,000 bacterium or cells per ml of bacteria indicates no infection, but the presence of more than 100,000 cells per ml of urine indicates a urinary tract infection. Remember that there should be no bacteria in the bladder or the kidneys themselves. However, bacteria can be introduced there, and that is what a UTI is, urinary tract infection. Remember that we inoculated two different types of plates, a CNA plate and an EMB plate with bacterium from urine or urine samples. So let's discuss what a CNA plate involves and a EMB plate as well. Recall that a CNA plate is known as the Columbia nalidixic acid plate. The selective agent in this plate is nalidixic acid, which inhibits gram-negative growth and allows gram-positive growth. The differential agent is blood agar in order to visualize hemolysis. And do you remember the three forms of hemolysis? Alpha hemolysis, where the agar turns a blue, purple, brown color, almost a bruise coloration around the bacterial growth. Beta hemolysis, which looks like a clearing around the bacterial colony, like a halo of bacterial around the bacterial growth. And then gamma hemolysis, which looks like no hemolysis, where the bacteria are growing on the agar and the blood is unblemished. Next, we had the EMB test. This stands for eosine methylene blue test. The selective agent here is methylene blue, which inhibits gram-positive growth but selects for gram-negative growth. The differential agent is eosine, which turns purple if there is lactose fermentation. All right, so with regard to the API 20E rapid test, the disadvantages, remember there are disadvantages associated with this strip. The investigator must use gram-negative raw bacterium. Also, the bacteria need to be pre-grown in lactose-containing media. However, the advantages are, that it's really fast and you get your results quite quickly. Also, do you remember how to get your 7-digit identification profile with the test? Each three wells on the test correspond to a number. So the first well corresponds to the number 1, the second well is 2, the third well, for example, is a 4. So what you would do is if whatever tests turn out positive, you would add those numbers together. So a negative, a positive, you would add 2 plus 4, so you get it a 6. A negative, and a positive, you would just add your, just number 4. If they're all negatives, then it's a 0. And this is how you get your 7-digit identification profile, which you would look up to identify your bacterium. Next, about bacteriophage lab. You should definitely know what a bacteriophage is. It's a virus that infects specifically bacteria. And remember, you used T4 phage to infect E. coli strain B, and you used Phi-174 to infect E. coli strain C. And what do you need to know here? One thing you need to know is, like I said, what is a bacteriophage? Another thing is, what is a plaque? And remember, plaques are lytic events. These are clearings in the bacterial lawn or in the pore plate where the virus has switched to the lytic part of its life cycle and lysed the bacteria. Remember, when you held these plates up to the light, they kind of look like Swiss cheese. There were clearings in the bacterial lawn. These bacterial clearings are called plaques. All right, now with regard to the soil lab, remember that we weighed out 1 gram of soil, did a serial dilution, and plated that. We plated on Saboraud dextrose agar or Glycerol yeast extract. What you should know is that Saboraud dextrose agar is for growth of fungi, while Glycerol yeast is a good growth medium for Streptomyces. And remember that Streptomyces are bacteria that produce antibiotics. They are bacteria that are plentiful in soil, and they even give soil the soil smell. Yeah, and fungi also are known for producing antibiotics. But they grow on the Saboraud dextrose agar. All right, for the oral biofilms lab, remember that we were studying a type of biofilm called plaque, but not plaques in terms of bacteriophage, but plaque on teeth. which is a type of quorum sensing event. We use 3 different types of media: Mitis-Salivarius agar for isolation of Streptococcus species, mannitol salt agar for halophiles like Staphylococcus and certain strep species, and MacConkey's agar for gram-negative bacilli like E. coli and other Enterobacteriaceae. And when we interpreted our results, right down here, for interpretation, remember that for mannitol salt, the salt-resistant staph species turn yellow. MacConkey's for gram-negative enteric rods, the coliforms, which are bacteria that are known as enteric bacteria, the coliforms turn red, and non-coliforms are clearish. And the Mitis-Salivarius agar, the small blue colonies are typically strep mitis. Large blue gumdrop-like colonies were strep salivarius, and the dark blue-black shiny colonies were Enterococcus faecalis. We can take a look here at some of these results of salivarius, mitis, Enterococcus. For the fungi lab, remember that we learned about the basidiomycetes, which are the mushrooms, the ascomycetes, which include Penicillium and Saccharomyces. Remember that Saccharomyces are yeast. Zygomycetes, which the representative here is the Rhizopus. I wanted you to know the structure of yeast cells. So Saccharomyces cerevisiae, remember these are the roundish oblong-shaped cells which divide by budding, right? They asexually divide by budding. Next, I wanted you to know about some of the structures associated with Rhizopus. For example, Rhizopus stolonifer forms a sexual spore called the zygospore, which kind of looks like this. And it can also form a sporangia, which is an asexual type of spore, which looks like this. Additionally, I wanted you to know about the structures associated with Penicillium and Aspergillus. With Penicillium, I wanted you to know what its conidia look like. The conidia spores, or asexual spores of Penicillium, look like finger-like projections like this, while the conidiospores of Aspergillus are more fanned out. They're more palm tree looking like this. For DNA restriction analysis, remember that we cast a 1% agarose gel, and then we added the TAE buffer, and to that gel we added our DNA to various wells. One well contained uncut bacteriophage lambda DNA, which is about 48,000 base pairs. To the next well, we added the same bacteriophage DNA, which was cut with the enzyme PST1, the restriction endonuclease, which cuts at palindromic sequences, right? These endonucleases, these restriction enzymes cut at palindromic sequences. PST1 cuts at a particular palindrome, EcoR1 cuts at a different palindrome, and HIN3 cuts at yet another palindrome. And then when we loaded these into the gel and applied a electrical current, remember we set the power supply to about 100 volts, then the DNA ran towards the red end of the electrophoresis machine. Remember, DNA is negatively charged, and so DNA will flow from negative end towards the positive end. We also used a loading buffer. Remember the loading buffer, which contained 2 dyes, xylene cyanol, which travels at about 2,000 base pairs, and bromophenol blue, which travels at about 300 base pairs. So we used the loading buffer as a marker for the progression of the DNA down the gel. We also used the loading buffer because it contains sucrose, which weighs down the DNA. Remember, the DNA went right to the bottom of that well, and it's because it was weighed down by that sucrose. I think that's about it. And then afterwards we stained this. Remember, we stained our gels with methylene blue, because methylene blue is a basic dye with a positive charge. DNA is negatively charged. The methylene blue should stick to the DNA. And then we destained and we took a look at our bands. And remember, the bands are indicative of different fragments of DNA. Smaller fragments of DNA run faster on the gel. Larger fragments of DNA run slower on the gel. All right, and the last module was serology, which makes use of antigen-antibody binding. One thing you need to know is that the monospot test for the Epstein-Barr virus is an example of an unknown antibody test because you're searching for the antibody, whereas the DIFCO staph latex test and the Clearview test for strep antigen, these were both examples of unknown antigen because you're looking for antigen. All right, let's go into a little more detail about the monospot test. Remember, the monospot test was a test for the Epstein-Barr virus. The Epstein-Barr virus can cause mono, the infection mono, or the disease mono, I should say. Infection with Epstein-Barr virus results in production of a heterophile antibody. You need to know what a heterophile antibody is. Heterophile antibodies are antibodies that attach to antigens other than the one that caused their production. Remember, not only will a heterophile antibody bind to the Epstein-Barr virus, but it will bind to, for example, sheep or bovine red blood cells. So the test, the serological test, has a reagent, which are these white latex-coated beads with antigen. There's antigen on these white latex-coated beads from red blood cells. A positive result would see agglutination, which means clumping of those white beads. If you see the white beads clumping, this means that the sample has the antibody in it, and the antibody is causing these beads to clump up because the beads have the antigen. Next, remember there are two unknown antigen tests. The DIFCO staph is one of them. Let's go ahead and start with the DIFCO staph latex test. For S. aureus, this is the S. aureus test. What we are looking for here is protein A, which is an antigen on the surface of the staph cell. We want to see if that is present. So to do that, we have blue latex beads this time, blue latex beads coated with antibody against protein A. So if our sample has staph with, Staphylococcus aureus with protein A, this will cause the agglutination, and you're going to get these little agglutination. You're going to get these foci of blue clumps for a positive result. And a negative result, you're not going to get these foci. Then there was the Clearview test for strep antigen. Remember, this was a test with strips. The strip is pre-coated with anti-strep antibody. Another antibody is attached to gold. This is your marker. You then swab a sample from the patient and then add that swab to the extraction buffer. The antigen moves up the strip via chromatography until it reaches the antibody that's already on the strip, and it will attach to this strip because it's attaching to the antibody on the strip. So now you've got the antigen attached to the antibody, and then the second antibody will attach to the antigen, the one that's attached to gold. That's the one I'm referring to. A pink line will form due to a reaction with gold. In this test, remember that one pink line is negative for strep, 2 pink lines is positive for strep. And with that, believe it or not, we reached the end of the Practical II review video. Hopefully this was informative. As always, I tried to remember a lot of the important details and notes that you need to know for the practical exam. However, please study everything. Go through on your own and try to find something I may have missed. I tried to touch on everything I could recall, but it is not a comprehensive review. It is more a review of the important points that I could recall. Please go through and check your own notes for anything I may have missed out on, but I think you can do it. Study really hard, study these concepts, and let's see how we do.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr. D.